we previously met, not so many times, but we did. And I'm very happy that you've found the time to help me out with my with our Batten Lab challenge to find awareness for Batten disease at the upcoming. Well, there's still 70 hours to go, I think. And with you, people may not know that, but you are an expert, in my opinion, on lysosomes. So I would like to talk to you, since Batten is one of the lysosomal storage disorders, a subgroup within the metabolic diseases. I think it would be really good to discuss actually what's happening if you look at it from a lysosomal perspective, and your perspective of the things that we know now and how that could be used towards treatment.
Yeah. Well, thanks a lot.
Yeah.
And it's a pleasure to be here. And I didn't tell you, but I actually saw you interacting with some kids in the hospital last week, and it was very inspiring also for me as a fundamental researcher to see you in action with your patients. So yeah, it's a very nice to be here and contributing to the challenge that you have actually ongoing. Well, as you said, I'm Nalan Liv from Center for Molecular Medicine, working on lysosomes, and they're my poison. I like thinking about lysosomes and how we actually improve therapies for all disorders related around lysosomal dysfunction. Well, maybe to start to talk about a bit what a lysosome is, right? So
I think that would otherwise be my first question. So I'm very happy that you just see that, because we're talking to people that probably hardly know what a lysosome is, and so really go back to the basis.
Yeah, let's do that. And I think the easiest way to explain it, at least this is how I explain it to like people that I interact, like, inside each one of your every cells is a very tiny but very hard-working recycling center, and this is what we call the lysosome. You can think of it as like a cleaning crew, a waste manager, but also a recycler, but all in one. And that's not even all, because it's also a traffic controller, energy regulator, and even a decision maker for the cell fate, right? So how it will use the nutrients that it gets from outside.
Okay, so you're very fast, and I think it's good to sort of consolidate what you just said. So what I hear you say, and now I'm memorizing it. So you say it's a dustbin on one side, but and so it's a recycling machinery. So it's just not throwing it away, but also reprocessing that into things that might be useful again to reuse in the cell. And but it's also I thought I heard you mention it's an energy sensor or something that relates to energy, and perhaps you can tell a bit more about that. And also there were some other things. So you see, this is how it works if it goes so fast.
I will definitely make it well slower, and maybe also even put in more analogy there. As I said, it's an energy regulator because, of course, when we think about energy, it's more the mitochondria that we think about, but lysosome is actually the unit that processes the incoming nutrients. And actually, it is the place in the cell that decides how it will utilize the available resources best. And depending on how much it has, or maybe not, it will either decide that the cell stays in a bit of a easygoing, resting state, or if it has a lot of nutrients available, it will go in the state to function fast and use these nutrients maybe to grow or move, or depending on the cell type, to perform more of its function. Maybe even easier to say, like, in a macro scale, right, it will be more easily analogized to the stomach of our cells. So just like our stomachs, they will break down incoming nutrients. and then they will make the broke down material available for other functions, available for other functions like our body is doing. So for, I don't know, circulation of our blood, even we need energy, and for that we need incoming nutrients. And lysosomes is actually good to analogize with your stomach because our stomachs are functioning in a very low pH, right? Sometimes, unfortunately, this is why you have this heartburn feeling when something goes wrong. And in the cell, also the lysosomes are functioning at a very low pH. So they are actually balls of acid. That is their trick to break down the incoming material. If they can't manage this, if there is a dysfunction, right, for whatever the reason, but it easily causes metabolic disorders like Batten disease. This is what we are talking mostly about today, but actually many other lysosomal storage disorders. And even recent research demonstrates in the last 5-10 years that they have a super important role in the formation and the progression of neurodegenerative disorders like Alzheimer's, Parkinson, and as well as in cancer. And I really think, and this is what excites me most, that understanding the fundamental mechanisms that these tiny structures inside the cells, how they function and how their function is regulated, that could transform how we also approach and maybe even treat these diseases in the future.
Okay, so what you're saying is it's actually it's a small but very important part of the cell.
Exactly.
And it has a lot of analogies to what the stomach does to the body as a whole.
Yeah.
And within, so if you would transport, so you have this stomach-like thing in the cell, and it takes care that nutrients get allocated in the best possible way, so that if you would see it as your purse or something like that, or the money that you have every day, then can I afford to buy something nice, or should I focus on things that are really necessary, or should I perhaps reallocate and sell some of my stuff in my house to be able to go on living? Is that the sort of the analogy that you...
That is exactly it. Yeah, you can think about it like the purse or the savings account. Depending on what you have inside, you can either decide on indeed going and buying the next nice Christmas present for yourself, or if you don't have enough, maybe you should just focus on the essentials.
Yeah. Okay, that is in one hand gives it a even more broad role rather than just being a dustbin or a recycle station. Yeah. On the other hand, it also complicates a bit of the view of it as having lysosomal storage, like with the lysosomal storage disorder. So could you tell me a bit about what you see under a microscope, or what is actually there in a lysosomal storage disorder?
So in a healthy state, when you look at healthy cells or healthy tissues, what we see is that lysosomes are very fast organelles. So because of also the acidic environment that they can sustain, they basically receive material and they are so quickly and so efficiently degrading it and making it available as other nutrients. So when you look inside, you would more see degraded material. Remnants of some membranes, remnants of some molecules that was there once upon a time as a whole. What is going on in lysosomal storage disorders, and this is an interesting phenotype because this is a bit of a roof phenotype. It holds true for majority of them. What you see is that the lysosomes, for different reasons for each disease, but in general, they can't sustain this fast degradative function anymore. So they, in place, start to accumulate and store undegraded material. And this is actually where the names of the lysosomal storage disorders are coming from. They are storing materials that they should be rather degrading if everything would be going all fine and according to the plan.
So the thing that they can't degrade, that's the thing they will store, but and that may differ between the different diseases.
Of course.
But the fact that they store apparently has consequences that are similar in similar diseases.
Yes. because unfortunately, more that they accumulate material inside themselves, that the accumulated material also starts hampering their function. So ultimately, for majority of the disorders, it turns out to be a dysfunctional lysosomal phenotype, and this is what makes them also kind of similar looking to each other. And sometimes, I mean, you will judge much better than I do, but sometimes if you can't do a genetic signature assessment, even difficult to distinguish them from each other, because majority of the symptoms for all these diseases are quite similar, and a lysosomal dysfunction ultimately underlies them mostly.
Yeah, yeah, I think you're partly right in the sense that it's a neurodegenerative phenotype and that is a difficult word, but in the sense that you get like pediatric dementia actually, and you tend to get that for a whole range of lysosomal storage disorders. The speed at which it happens is really different between the different ones, and that is something that I've never fully understood why it makes such a difference. Is that something you could?
Maybe I can elaborate on it. Of course, we don't know exactly the reason what makes them also because we basically don't know the function of each and every gene and protein that is unfortunately causing the problems. But I think there are two things there. So most of the time, not with Batten disease, I have to say, but with some other lysosomal disorders, it is an enzyme that is in the lysosome, that should be in the lysosome, that has a problem. And if these enzymes mostly have specific targets that they need to degrade. So depending on what the target is, it can have lighter or heavier consequences, which would, I think, protest earlier or a little bit later, maybe in life. But also, it also defines which part of the body it affects more, right? So, as you said, majority of the time, it is first the neuronal system that suffers most, and that is most likely for multiple reasons. First of all, the neurons are interesting cells. They are very polarized. They also don't really divide and renew themselves as much as the other body's parts. But some lysosomal disorders actually have more a liver or a kidney dominant phenotype. So I can't say they're all also neurological, but depending on what goes wrong, a specific part of the body will be affected most or first. This is very unfortunate, but
so if I would try to make it as simple as so that I can understand, is that what you say is that the thing that is stored may be more or less detrimental for the cell, so that if some substance A gets stored, it may be much more problematic than another substance.
Exactly.
So that's one part, and the other part is probably due to the fact that some processes or some degradation needs to take place in certain parts of the body, and then if you have something that so apparently the neurons are very vulnerable. So regardless of what happens, usually the brain and other neurons, the neuronal cells will get damaged, but not always, but mostly, and other cell types may be less vulnerable.
Yes, for example, I'm thinking about an example in Gaucher disease. It will first protest more in the liver or the spleen or the bone marrow of the patient. But I personally think this is not because neurons are not affected at all. Most likely they are also affected, but it is much more of a fat problem, lipid-based problem. So we observe it first in the lipid-related organs of the body. That's the other thing, and majority of the kids or patients, let's say, they don't have a very long expected lifespan, so it is also rather difficult to judge if they would have also problems in other organs eventually, if we would be able to follow them up or not. But majority of the LSDs, I think we will be right if we say majority of them show first a neurological phenotype, and that's the neuronal vulnerability, as you said. But some of them will show more on liver or spleen, or sometimes in the immune system.
Yeah, I think we have to a bit move back to Batten disease. So is Batten disease very much akin if you look at it from a cellular perspective, or are there differences between if you look at it with electron microscopy or other visualizations? What are there also, apart from the analogies that are quite obvious, are there also differences?
There's definitely differences. And I think, well, of course, Batten disease is also a collection of genetic alterations that we refer to, and they phenotypically look similar, but inside the cell, they can also differ slightly from each other. What I can say holds true for majority of the Batten disease related mutations and tissues is that, well, you do see the lysosomal storage phenotype. So lysosomes get more enlarged, they get full with accumulated materials. But in some variants, you will see that less. They are more prone to having smaller but still full lysosomes. But in general, and especially the most widespread form of Batten disease, shows quite enlarged and full lysosomes. That is also interesting, right? Because the gene or the protein that we know to be related to the Batten disease, most spread version of Batten disease, CLN3, it's an interesting one because we still don't know the exact function of it in the like how it contributes to the proper functioning of the lysosome and what exactly goes wrong when it goes wrong.
Yeah. Now you say something that there was a paper in Nature a few years ago, maybe 2-3 years ago, by a group in Stanford, by Monter, and he claimed that he had elucidated the role. Are you not completely agreed with that, or is it more that you say, well, there's probably something more to be able to fully understand?
I think that what they elucidate is very neat and very nice. I'm not sure if it is the full picture. That is what I feel around it. Also, when I look at as a cell biologist, when I look at the structure of the protein, when I look at how it makes its way and where it should function and how it functions, I am fairly convinced that there's a lot more that we can and we should find out about this protein and its exact role so that we can also approach Batten disease in a much more accurate way.
Okay. So in that sense, should we do more research? That should also, this would be an important path to explore to understand it even better. And do you think the lipid-like, I mean, lipids, everybody knows about lipids, I mean, fat in other words, but lipids actually are a whole bunch of different molecules, and if you zoom into that, it gets really complicated. And the specific lipids that this group showed, do you think this methodology that you can address that could also help get a step further to really a full appreciation of what's happening?
Yes, so I think you're referring to the method that they actually analyze the content of lysosomes upon after mimicking the disease mutations. Definitely. And not only for this disease, but also for other lysosomal storage disorders. Because of course, on the side, we are developing enzyme replacement therapies, right? Gene therapies to correct the underlying genetic or phenotypic effect. So for example, for some lysosomal storage disorders, there is actually enzyme replacement or therapies that helps to degrade and get rid of this accumulated material that we are talking about in the lysosomes. So yes, in that sense, it's a very elegant approach if we can identify what is exactly getting accumulated per disease or in this certain disease, for example, in Batten, we can come up with a drug that would get into the lysosome and degrade that set of lipids in this case, but whatever is getting accumulated, and maybe elevate the symptoms of the patients.
That sounds really neat, but also like something that is hard to attain. Is that am I making it too difficult?
No.
No, it's difficult.
Well, no, it is of course challenging, but I think this is so as I said, it does, we know that it does work in some lysosomal storage disorders already. So there's always I think it is definitely worthwhile to invest in these studies because the potential therapeutic breakthrough can be enormous. Again, you would know these numbers much more than I know, but what is it? At the end, I think nearly like 1 in every 7,000, 8,000 live births results in a child diagnosed with a lysosomal storage disorder. And these conditions are often quite severe conditions. And it's, and that only underscores the urgency of why we should research more into this field and invest in all the ways that we can in finding better therapies, understanding what is exactly going wrong in these disorders.
So it sounds really as if there's a lot to learn, also a lot of interesting research questions that are there. I know that if you look at people working on lysosomal storage disorders or even metabolic disorders more broadly, there are not so many. Is there, I mean, while the questions are really interesting, I guess there's many people that find it interesting as well. So what makes that sort of an underexplored territory?
I will give one answer, which is I think more broad, or because I actually don't know. I mean, I'm with all my heart want to invest, I think, for the rest of my life in this research. But I think there are two things. First of all, the lysosome is not a very easy organelle to study. It is a very dynamic system. So we are talking actually it is like one per cell, right? Like lysosome, tiny structure in a cell. But there's approximately 100 to 300 lysosomes in one of your cells. And it is also doesn't function as a solo system, but it functions as a part of a complex network of organelles that we call as the endolysosomal system. So collectively, they decide on what to take up, how to sort it in the cell, and then either to degrade it or not, and so forth. And that dynamic system makes it always a bit, I mean, it poses a challenge for methods also that you can use, and for example, also even for microscopy methods that we can use to visualize them. Okay. And it's also very small, right?
Yeah. But so it is small, and then it's dynamic, and because it's dynamic, you cannot really say, well, the lysosome is here, because at the next moment it will be somewhere else, and it will be a different form, and it will have changed over time. And that, really sort of having a standstill and being able to look at the lysosome, it's actually something that is happening. It's a movement in a way.
Yes.
So it's almost like physics, right? So that that you can't really say that something is there at the position and be precise about its role or something like that. I don't know. I'm paraphrasing it in a wrong way. But that seems like hard. But hard could also, usually scientists like hard stuff.
Yeah.
As they think they can cope with it. I was guessing, but perhaps that would be a next point, that there's also a money thing that surrounds these.
This is indeed the next thing, and I will link it also what we just commented. As I said, like collectively, like the lysosomal storage disorders unfortunately affect many lives, and also, as I said, we just because of the reasons we mentioned, like they were also traditionally hard to study and understand. Now we are getting there. we are just trying to understand there are also significant roles in neurodegenerative disorders as well as in cancer. And of course, if you say cancer, it is rather easier to raise money and find the money to do research for it. It has been traditionally the case because a lot of people's lives are affected with this disease. Well, the challenge here is that when you say name a single lysosomal storage disorder, sometimes it's not a broad group. And whereas actually the lysosomal storage disorders as a collective set affects so many lives. So I think it's also the lacking awareness that we all have in that perspective, like how many, like how widespread the lysosomal storage disorders and also the metabolic disorders that are caused with problems around the lysosomes are compared to other diseases which are much easier to raise some money for, right? I think it is, we all know, I mean, there's a lot of research going on into Alzheimer's, Parkinson's, cancer, different forms of cancer. But this is also a field where, well, it is rare diseases, but collectively they are not rare at all. And we should raise awareness about this.
Yeah. Okay, and so this is actually why I'm trying to keep myself awake for 24 hours and spend time on this challenge and create awareness. So, do you have? So I think I heard an advice about making this more happening, and that is to have some bins like of information. So by not calling it, let's say, GM2 gangliosidosis or CLN3 type, CLN type 3, but to call it lysosomal storage disorders, and to focus more on the broader content. From a so I that makes sense from a lot of perspectives. The only thing is that if you would look at what happens to patients and you want to make sure what happens to patients, it's not the same for this whole group. Yeah. So one of them may have something completely different, skeletal problems, or as you said, problems of your liver or your spleen. So then if you would, but I can see the point that it's still, even while there are differences, it would still matter, like what is being done now with metabolic diseases, to just make it a larger bin.
Exactly, and we will get to understand more and more. This is also what part of my research is lately also focused on. Lysosomes actually have a very significant role even in many cancers, in the formation and progression of many cancers. So well, I really think we should focus on metabolic disorders as a separate bunch, but also we should benefit from the research, fundamental research going around lysosomes in a cancer related, I don't know, study, as well as a neurodegenerative related study. And yeah, but this information will come all together, and I think at the end we should, it will beacon the hope for future therapies for a lot of these disorders.
Yeah, and do you see ways to sort of combine these two fields? So it would I can imagine if you ask for money, it's not a simple thing, but it would be nice if you would have let's say a larger brother, so that the larger brother, let's say cancer or diabetes or bigger disease, cardiovascular disease, they sort of embrace the super rare ones and try to, while getting this money, also sorting things out for the small kids out there. Yeah. Do you envision that would be a way to go?
It can be, because also it's also I don't know, even some of these disorders are symptoms are defined as childhood Parkinsonism, for example, right? So why not?
Actually, what you see with CLN3 disease, it is Batten disease. It's very much like that. To be clear, CLN3 disease and Batten disease are usually called this two names for the same thing. Yeah. So sorry, but I was interrupting you. You were saying something.
No, this is exactly it. Like, I think, as you say, like bigger brothers or bigger roof umbrella. For example, I think neurodevelopmental, neurodegenerative disorders, as well as the rare disorders, rare lysosomal storage disorders that affect the function of neuronal system, they can all support each other, and this can be part of it partly. But then this also, and then again for the liver targeting disorders, then maybe it will be benefiting indeed to put them in this or evaluate them in a similar system with diabetes, other like metabolic syndrome research. What is going on in the processing of incoming sugar as well as fat molecules? Because that is a common theme. What goes wrong on the cellular scale there?
Yeah, and so you're also saying that that we should try and make use of the fact that even scientists that work on completely different diseases, they may also be knowledgeable on aspects, since they are also studying cells.
Exactly.
And what happens in cells, regardless of, and that's also where things can become seem very costly. But because it's not, if you solve one of the 1900 diseases, I can imagine if you so a lot of research for this one in 1900. But I think it can sort of be used in so many others that the next one will be much easier.
Yes.
Much like recognizing the first one, or do your first math chore and doing it for the first or the fourth time.
Yeah, like this is the power of fundamental research, right? It's like you invest. It is hard to visualize what will the turnout will be. It's a little bit in that sense, maybe higher or longer term investment. It is like, I don't know, investing in education, right? So it's not something that you see the immediate effect, but you see the effects. There's certainly, and like assured, there will be an effect in the future that you get, that you benefit from it economically and so forth. And I think it's a little bit the same here. If we invest more in understanding fundamentally how a lysosome functions, and all these proteins that are actually mutated, or sometimes even not at all in the picture anymore in the cell, in these diseases, what are their exact function? If we invest a bit more into this, we will be benefiting a lot of diseases at the same time, and a lot of clinical problems at the same time.
I heard another advice coming past, and that is the comparison with education. So actually what you're saying is with education, you don't ask whether that's useful, and perhaps understanding cells or doing science to understand things, you shouldn't ask why that is necessary. Just like, because we know it has a solid base, that it has always been useful, just like education has always been useful.
Exactly.
I think that's also a good ending point of our meeting. Thank you very much for all the insights you've been
Thank you for inviting, and it's a pleasure to be part of this.